This project is growing algae from fertilizers. The main issue associated with the project is pollutants from yard waste on the coast. Living in Florida, there are many homes built near drainage systems or directly on the water. These drains are transporting pollutants into the ocean and causing algae to bloom. One polluting factor is from mulch, dirt, or grass is fertilizers. Algae blooms and red tide are effects from these polluting fertilizers. While algae is necessary for the balance of the food chain as a primary producer, they can cause destruction if they overpopulate and become out of control, which is essentially red tide. Then health concerns arise from red tide involving animals, sea life, and human respiratory systems. Yard waste such as mulch, dirt, and grass contain fertilizer contents on them. These fertilizers can be extracted from the surface and used to demonstrate the cause of the blooms. Fertilizers are harmful because they contain nitrogen, which speeds up the eutrophication process, which is the excessive plant life growth that causes the loss of animal life. The algae will be grown using a medium from anabaena algae. This will mimic the real growth of blue-green algae in drains. It's known that phosphate and nitrogen are harmful for algae blooms in the ocean. By studying and understanding this effect, different types of waste are deposited into the drains and can be tested. Sarasota has a drainage basin, which they call the Sarasota Bay watershed. And according to the Sarasota Water Atlas, there are 161.4 square miles of this watershed containing 153 lakes, 34 rivers, and 11 bays, all intertwined between the modern houses and businesses and industries such as that. The pollution collected from these drains is excessive and needs a solution. Knowing which type of yard material may help limit this used in agriculture. An article published by Penn State Extension says that grass clippings can provide up to 25% of a lawn's total fertilizer because each grass contains 4% nitrogen, 2% potassium, and 1% phosphorus. Soil, specifically topsoil, contains about 40 milligrams per liter of nitrogen, and wood chips have actually seemed to deplete nitrogen levels in the soil. These excessive amounts of nutrients can cause the overgrowth in algae over short periods of time and cause algae blooms, which severely impact human health, aquatic ecosystems, and the economy. The focus is the anabaena blue-green algae. The methods used to conduct this experiment start with growing the initial algae in an Erlenmeyer flask and adding sucrose and ammonium hydroxide to promote the growth of this algae and the steady increase. Then once the algae reaches 0.17 AU measured with a spectrophotometer absorbency, it's mixed 100 milliliters of the algae and 100 milliliters of the steep solution. The steep solution of grass, dirt, and mulch is 40 grams of each variable with 400 milliliters of DI water, which sat for 25 days, then was filtered out with coffee with a filter. And then it was placed together in a windowsill for several weeks, taking the absorbency each weekday for 5 days. The second trial was set up with an additional DI and medium culture and measured the data the same with a spectrophotometer. And the first trial, as you can see in the graph, the grass clipping solution promoted the most growth in algae culture. It started out thicker and darker with promoting more algae growth. It started at 0.42 AU and grew to 1.14 AU, which is a 0.72 difference. The mulch had a 0.50 AU difference, and the dirt had a 0.44 AU difference. The grass also had a consistency growth within the whole experimental trial. And continue with this project, trial 1's algae is going to be put through a filter pump to see the dry mass. Then trial 2 will continue data collection, and the trial 3 will be set up using the same DI water and medium as trial 2. And each trial, the testing and data observation will be finalized roughly by January 10th, if not sooner, for the consistency and accuracy using the spectrophotometer and the pump for the dry mass. However, as of right now, the preceding data collection of the grass clippings and are seeing the most growth, even in trial 2, which started at 20.24 AU and is already at 0.90 AU after 7 days. So as of moving forward, I hope the grass should see the most growth in each trial. These are my references.